Think of the human immune system as a massive biological anomaly detection network. Right now, your T cells are constantly patrolling your body, latching onto cells and scanning them for mutated proteins, signatures we call neoantigens. For years, blockbuster cancer treatments like immune checkpoint blockades have operated on a relatively simple premise. They essentially remove the immune system's safety brakes, assuming that once the restrictions are gone, the patrolling T cells will automatically spot the tumor and attack. But if a patient's immune system doesn't already have T cells trained to recognize the tumor's unique signature, taking the brakes off accomplishes absolutely nothing. The T cells will simply float right past the cancer. To fix this, we have to manually upload the tumor's exact coordinates into the immune system. We start here, using next-generation sequencing machines to map the tumor's DNA and catalog every single mutation it possesses. A single tumor can easily produce thousands of unique mutations. Finding a cure becomes a massive data filtering and prediction problem. Modern oncology has evolved into a problem of feature selection, isolating the specific data points hidden in that genetic noise that will trigger a systemic immune response. The old way of predicting which of those thousands of mutations to target relied on measuring physical fit, a metric called binding affinity. Scientists would look at a mutated peptide and try to calculate if it could physically wedge itself into a cell's surface protein, known as an HLA molecule. This physical fit model was notoriously inefficient. Researchers would apply a standard 500 nanometer binding cutoff, yielding hundreds of predicted targets, only to find that a dismal 6% of them actually stimulated T cells in the real world. To solve this, researchers use next-generation machine learning. These algorithms train on massive datasets and functional T cell assays to improve prediction. The model accounts for biological blind spots. If a patient's T cells ignore a mutation, it filters it out, outputting only viable targets. The algorithm also prioritizes the hierarchy of the cancer. It specifically hunts for foundational or truncal mutations that drive the tumor's growth, rather than newer branch mutations. Target the branches, and the tumor will simply evolve around the attack. Superior machine learning isolates these foundational driver mutations, targeting the core genetic alterations the tumor cannot shed without losing its ability to grow. Once the algorithm selects the top 20 to 60 targets, that data is compiled into a custom mRNA payload. This payload is packaged into lipid nanoparticles, or LNPs. These microscopic fat bubbles serve two functions. They keep the mRNA intact, and they act as native adjuvants. An adjuvant is an immunostimulant. The LNP's chemistry triggers localized inflammation. This forces the immune system's attention to absorb the nanoparticles and execute the uploaded mRNA code. Crucially, the code targets CD4-positive helper T cells. Inside the tumor microenvironment, these helper cells recruit macrophages to dismantle the cancer. They also shut down the tumor's countermeasures by actively driving down protective regulatory T cells, acting as system administrators, granting execution privileges for the attack. The final mRNA lipid nanoparticle is an executable software patch explicitly engineered to override localized tumor suppression. Even flawless code can fail if deployed into the wrong environment. In late-stage cancer, the tumor has already established a highly corrupted immunosuppressive fortress filled with exhausted T cells that can no longer mount a fight. To bypass this, oncologists are shifting to a deployment strategy known as the adjuvant setting. Here, the vaccine is administered right after surgery. The goal is to hunt down microscopic, unseen residual disease before the cancer boots up its robust defense systems. This line graph illustrates the results. In clinical trials from BioNTech, Moderna, and Merck, focusing on melanoma and pancreatic cancer, patients receiving these custom vaccines show massive T cell expansion and significantly prolonged recurrence-free survival compared to standard care alone. Clinicians are moving away from broad, generic chemical toxins that rely on systemic toxicity. Modern oncology operates as a rapid response data pipeline, sequencing the tumor, predicting its vulnerabilities with machine learning, and using custom mRNA code to program the patient's own immune system to recognize and eliminate them.